Like to describe our work where we improved block fitting and machine learning methods that analyze AcidoCEST MRI. I have no relationships to declare with regard to this. AcidoCEST MRI is a method where we use iopamidol, which has an amide proton that can be selectively saturated, and when that proton exchanges with water, the saturation has been transferred to the water, lowering the water signal. That can be seen in this experiment where we have iopamidol in the inner tube relative to no iopamidol in the outer tube. And if we selectively saturate frequencies, eventually we get to 5.6 ppm, the resonance of this proton, and we see the CEST effect where the inner tube goes dark relative to the outer tube. This happens at 4.2 ppm selective saturation as well for these other protons on the agent. If we saturate directly at 0 ppm, you can see the inner tube and the outer tube have direct saturation of water. We collect a symmetrical spectrum so that we can actually analyze the data in a variety of ways. We prefer to fit the CEST spectrum with the Bloch-McConnell equations that include the term for chemical exchange. Now, one of these protons actually has a decreasing CEST effect as the pH increases, while the other proton has an increasing CEST effect as pH increases due to the difference in chemical exchange rates of these protons, which can be used to estimate pH using a relatively simple relationship. Again, the Bloch-McConnell equations are a better method for analysis. We are basically watching glycolytic metabolism within tumor cells, the conversion of pyruvate to lactic acid, as seen in a variety of preclinical in vivo studies that we've done over the years. So we did a project where we took a series of samples at 8 pH values, 5 concentrations, 5 T1 values by doping with gadolinium, and then performing CEST at 5 temperatures, 6 saturation powers, and 6 saturation times. So a lot of CEST spectra, along with their corresponding T1, T2, B1, and B0 maps of these samples within these boxes. We found that we are underestimating the pH only by 0.03 pH units, which is about the accuracy of a pH meter. So we're doing about as good as we possibly can relative to the gold standard pH meter. We do not need other T1, T2, B1, and B0 data to get the best accuracy and precision from the CEST fitting method, the Bloch-McConnell fitting method. And fixing parameters during the fitting to the experimental values does not improve the estimate of pH. However, it will improve the calculation speed as which is intuitive, of course. The saturation time can be as low as 2 seconds. Saturation power at 3 to 4 microtesla produces excellent results. As a problem, this technique does require at least 15 millimolar to have a reasonably good accuracy and precision, although you can get a measurement with less accuracy and precision if you have lower agent within the phantom or the tumor voxel. It is temperature dependent, but as long as you keep your mouse 37 degrees plus or minus 1 degree, you'll have a very good pH measurement. So then we took this very large database and we took 70% of it to train a random forest regression model. And then we tested the remaining 30% to see how accurate and precise we were. And we had outstanding accuracy and precision for measuring pH within the whole physiological range for tumors. We take subsets of this data, not the entire part, but subsets at different saturation, time, and power. While the errors are going to be larger than what's seen up here, but this graph or this figure here shows that the sweet spot for saturation time is about 3 to 4 seconds. Saturation power about 3 microtesla, and not too different than what we've shown earlier. We do lasso regression. This is an inferior technique, and we get higher errors. So we recommend the random forest regression works just fine. So to summarize, we can estimate pH with block fitting. You do not need extra MRI data. We have optimal conditions for the acquisition time that for this particular analysis method. Minor temperature dependence, but that can be controlled quite easily. And then finally, the problems still remain that you need a lot of agent and you need a lot of time to calculate a pH map. Machine learning with random forest regression also has great precision and accuracy, optimal acquisition conditions. Surprisingly, we've built temperature into this model, so it's temperature independent, which is a minor advantage, but not that critical really. You do require extra MRI data beyond CEST MRI for best fitting or best estimates of pH. And that's because machine learning is using a multidimensional approach to identify the pH here. It requires less agent, which is a very large advantage. And then also it's fast, only requiring seconds to estimate the map once the model is built. So with that in mind, block fitting doesn't require more experimental data, but it will require more agent and more analysis time relative to machine learning, indicating that machine learning is an outstanding method to be translated to the clinic. And then finally, I'd like to thank Tianzi Li and Julio Cardenas Rodriguez, two outstanding researchers I've had the pleasure to work with for many years, who are experts with CEST MRI and machine learning. Thank you.